Welcome to Brain Ponderings, your window into the world of neuroscience research. I'm the host, Mark Matson, and today it's my great pleasure to welcome Alex Kwan. He's a professor at Cornell University, and he'll have interesting background that I can have him talk about. It's kind of a combination between, well, physics and neuroscience, I guess. And recently, he's been doing some really important work on trying to understand how, why it is that psychedelics seem to have long-lasting effects, for example, on in reducing depression, anxiety, and as he'll talk about, there seems to be some structural changes that are going on in certain circuits in the brain. So welcome, Alex.
Yeah, glad to be here.
Okay, so can you talk about your background? Go way back. Where'd you grow up? Where'd you go to college? And we'll go from there.
Okay. So I grew up in Hong Kong. Hong Kong. I was born in Hong Kong. Um, I uh, I did my elementary schools there, and then by 12 years old, like many people in Hong Kong, my family immigrated to Canada.
Oh,
After that, I continue high school up until undergraduate in Vancouver in British Columbia. Um, for PhD, at that time I was studying engineering. I also very interested in physics. At my undergrad university, which was Simon Fraser University in Vancouver, BC, they have a program engineering physics, and that's what I was doing. Mostly electrical engineering, but also, a little bit of condensed matter physics.
hmm.
Then for PhD, I was looking actually into the physics program, and I want to do more. I was very interested in superconductivity at that time. Cornell is quite strong in that area. So then I decided to go to Ithaca, New York, to do my PhD. But then, sometimes you get these opportunities, I think, in life. That part didn't quite turn out in terms of studying superconductivity. Within 1 year, I switched my direction to biophysics. I ended up joining a biophysics lab at Cornell. That was Watt Webb's lab, and he's very well known for building optical microscopes.
Hmm.
So, for example, he invented two-photon microscopy with Winfried Denk and others. So I was in the lab in my PhD building optical microscopes. These microscopes turns out to be very useful for studying biological samples, particularly in the brain, which is very scattering. And then I got very interested in neuroscience at that point.
Alex,
So
I should tell you, so
yeah.
my
Uh-huh.
postdoc work, I was studying how neurotransmitters regulate neurite outgrowth and synaptogenesis. And I discovered that glutamate plays an important role in the wiring of neural circuits in the hippocampus. And this is what in the 1980s, and Roger Chen had just come out with the FER2, which is a calcium indicator dye. You can
Yeah.
look at neurons, and by looking at changes in fluorescence, you can get an
Hmm.
indication of the calcium. And so my postdoc mentor, Stan Cater, he pointed out to me that actually, confocal microscopy, so the big value of it is it eliminates blurring, essentially. You can get crisp images. So turns out you can just take a regular microscope and cut a slit in a piece of paper and slowly move it across uh, well, the objective, the microscope, and you can get a clearer image because you're only exposing the area in that slit to
Oh,
the ultraviolet light at any one time.
like a structure illumination. Yeah.
Yeah, yeah.
Uh-huh. Yeah. No, I use those dyes. I've the one I used the most in PhD was Flow 3 and organ green bapta. So I'm back at that stage, and I think these days people get spoiled with these really sensitive and bright calcium dyes. But yeah, I studied them all the way back to my PhD.
Yeah, now they have genetically, you can uh, put genes in neurons that allow you to look at calcium levels only in the neurons that you put those
Yeah.
genes in.
So I mean, so that was my PhD, and then for postdoc, I decided I really want to become a bona fide neuroscientist. So I went to Berkeley to work with a visual neuroscientist, Young Dan, which got me really into cortical circuits. And then my own
Hmm.
research direction switched to drug only started when I have my own lab, and I have my first faculty position in Yale psychiatry. That department was chaired by John Christo, who discovered ketamine as antidepressant.
Uh,
So when I was hired there as a basic neuroscientist, I thought I should do something related to the department. And then we thought, oh, everybody is studying ketamine, so maybe we should study a little bit about ketamine. I wasn't too interested at that point. I was still very interested in just cortical circuits and the basic biology of it. But I thought I should do something, closer to what other people are doing in that area. So we started looking at ketamine's effect, but that just got more and more interesting. And I feel there's not a whole lot of, really good kind of basic systems neuroscience done for these drugs. And then over time, I've had my lab now for 12 years, and we just grow more and more interested in drug action in the brain. And so that's how we got here.
and how are you looking at the function and structure of neural networks?
I mean, with my training, a lot of what we do is still optical imaging. So
Okay.
we do a lot of laser scanning, microscopy, specifically two-photon imaging, which give us really high spatial resolution so we can see individual neurons in the brain and
In
look at
vivo, in
in
living in,
vivo.
yeah,
Yeah, that's a big part of our research, which is these are in live mouse brain.
yeah.
Most of them are awake, sometimes anesthetized. So the neural circuits are still intact, in the brain. But we have enough resolution to look at not only single cell, but also single dendritic spines, which would be the putative connections. We can track them over days. Although recently now, our lab also expanded into doing quite a lot of electrophysiology as well. So we also do a lot of just recording firing activity of neurons. So now we, incorporate other tools to study drug action, but still a lot of imaging.
Okay, and then um, talk a little bit about, just go a little bit background on psychedelics, just to get everybody up to speed, and the convergence of their mechanism of action on seems like one important signaling pathway.
Yeah, so I mean, we got quite interested in psychedelic, or more specifically, classic psychedelic, I think about 5 years ago. Really inspired some early work out of the UK, but also out of Johns Hopkins,
Yeah.
where they show that a single dose, or in some of those early study, two doses of psilocybin, usually with psychological support or in an assisted setting, can really dramatically reduce depression symptom and sometimes also have very long-lasting effect. So that's quite surprising clinical finding, right? Because the drug itself has known subjective effect, but those last only 4 to 6 hours in human after oral administration. Yet some of these benefits for mental illness seems to last for weeks, if not even months. So we were very interested in biological, what these drugs do. And then my lab study animal models like mice. One of the first thing we asked is, do these drugs also have some kind of plasticity uh, uh, facilitating type of role? This is mostly due to our knowledge of other drugs. So before going to psychedelics, my lab also have studied, as I mentioned, ketamine. And ketamine, one of the unusual action is that it really induces plasticity of synapses in the frontal cortex. So that was really the first study, and still driving a lot of our research to understand if psychedelic could also drive some of these plasticity in the brain.
And so ketamine, its action is on a certain type of glutamate receptor. Glutamate's the most prominent and important neurotransmitter in the
Yeah.
brain. And so this NMDA receptor, glutamate receptor, is a calcium channel. So when it opens, calcium goes from outside the cell to in, and calcium is a very important signaling uh, signal for a
Hmm.
lot of things, for both short-term and long-term memory and a lot of plastic structural changes. Um, but psilocybin from the mushrooms, it its target is not an NMDA receptor, and yet there's some overlap in the and I guess the psychological effects and even clinical effects of the two. What are your thoughts about that?
Yeah, I think that is one of the big mystery for these drugs. You pointed out some similarities in terms of if you compare just ketamine and psilocybin, which is a classic psychedelic. They clearly both have acute behavioral action in terms of some subjective action. I would say there's some nuances, right? Ketamine is more thought of as a dissociative, and then psilocybin more of a visual perceptual change, although there's also cognitive and emotional aspect. But they both seem to be then useful as a treatment for mental health. Um, yeah, and then at the biological level, there also seems to be differences and convergence where clearly there are different targets in the brain in terms of the receptors. For psychedelic, it would be the serotonin receptors
Yeah.
that it primarily engages. Yet in our study and other people's study now, there also seem to be similarities in terms of how they engage plasticity, which is how they modify the synaptic connections in terms of the area that they engage in, areas like the frontal cortex, the amygdala. There's some common area that they share. Happy to talk more about that. Um, and then, yeah, ultimately, also even some of the behavioral effect in animal models. So it is quite fascinating how you can start off with completely different target, but end up converging onto some similar downstream pathway at the circuit level.
Now, one thing that some people don't, well, neuroscientists know this, but the only way that neurotransmitters such as serotonin or dopamine affect behavior is by modifying activity in glutamatergic neural circuits. So the core circuitry throughout the cerebral cortex, hippocampus, etc., cerebellum, it's mostly excitatory glutamatergic neurons and then a small number of it local inhibitory GABAergic neurons. So I think, the general public, they hear psychedelic, serotonin, 5-HT2A receptor, and, they focus on the importance of serotonin. But the serotonin receptors are on glutamatergic neurons. And so one would think then it's likely changes in the glutamatergic neurons that are responsible for at some point they have to be responsible for any of the psychedelic experience or maybe even the long-term effects of a single dose. So can you talk a little bit about, you mentioned a few brain regions. Can you talk a little bit, say the prefrontal cortex, which is a brain region that's most highly evolved in humans. It's critical for decision making and a lot of cognition. So talk about the prefrontal cortex and its potential role in psychedelic effects.
Yeah, my lab, many of our study have focused on the medial frontal area of the mouse. And we focus on more the dorsal, like the upper aspect of it, which it's hard to draw exact homology
Yeah,
between different species,
yeah.
but I think one could make a case that it probably correlate most to the dorsal anterior cingulate cortex
Uh,
of the primate.
uh,
Um, so that's also it for the aficionado in a granular area in the brain that's really critical for interpreting rewards, like reward guided behavior, like how we go about our day. If certain things are more rewarding, we would do it more. Also, it's been implicated for conflict resolution. Like if you have several urgent item, like which one do you pick, or if a certain task can give you both joy and sorrow. Like should I do it or should I not do it? So, and then these regions both actually the dorsal, I think, and the ventral aspect of the anterior cingulate in human have been implicated in depression through various kinds of neuroimaging study. In the mouse, we know that these regions are important for the drug action, mostly because we also have done some brain-wide study. So in the mouse is, our lab and other labs have done where we would give the mouse a single dose of these drugs, whether it's ketamine or psilocybin. We've done both, and then look at activity-dependent gene expression, and then map where these genes are expressed, which give us an indicator
Uh,
which brain area is active.
yeah.
And we can now, with nowadays the technology, we can clear the brain and image the whole brain and look at, where these signals are. And this area in the sort of dorsal anterior cingulate cortex is one of the most robustly area that's lit up for both ketamine and psilocybin. But I will say, it's not the only area. We've also find substantial signals in central and basolateral amygdala. We've seen some in locus coeruleus, some in the amygdala. So it is these drugs and I think that to some extent the disorder engage a broader neurocircuitry, but definitely this sort of medial frontal area as part of the anterior cingulate cortex is a key node, and that's why a lot of our study have focused on that area.
And which neurons in the brain seem to have the highest numbers or density of the serotonin 5-HT2A receptors? And would that give a clue as to, well, which neurons might be most responsive to this?
Yeah, that's also a great question. I mean, to backtrack a bit, I mean, the serotonin 2A receptor is obviously important because if you block it in human, and then you take a psychedelic, then you would no longer have a trip or have the subjective effect. So whether that's also involved in the therapeutic effect, I think is debatable, but
Hmm.
for sure it's a key part and key target of the drug. Um, What's exciting about doing psychedelic
Yeah.
research these days now is that we have a lot of public database, for example, transcriptomics data that tell us where the receptors are expressed, at least at the transcript level. So I can tell you that the serotonin 2A receptor in the frontal cortex express quite strongly in the pyramidal neurons.
Yeah.
So they are present in both the so-called pyramidal tract neuron, which go from the frontal cortex out in the subcortical nuclei all the way down to the pons, but also in the intratelocephalic one, which is the one that form recurrent cortical-cortical loop. Interestingly, the 2A receptor also found in some GABAergic cell types, like the fast spiking basket cells, some layer 1 neuroglial cells, and then also some excitatory layer 6 neuron. So, but they're not expressed in all cell type, but select cell type in the frontal cortex have pretty high amount of the 2A receptor, which will make them prime target of the drug.
Okay, you've had several uh, outstanding papers come out just in the last few years on your work looking at structural changes that occur with psilocybin mainly. I think you had a Cell paper and a Nature paper.
Yeah, thank you. Yeah, last year
Did
was a
you
big
want
year
to
for us.
do you want to go through those just um, and kind of talk about a little bit of the details of how you approached it and what you found?
Sure. Yeah. So. Uh, Again, one of the early finding from the lab was that, and this was actually an earlier study we had 4 years ago, which inspired some of these recent study, is that again, if you give a mouse a single dose of psilocybin, it can induce the growth of new connection. And in that study, we assess it by imaging the brain and looking at the formation of new dendritic spines. And then we can track it over days and show that some of these spines can stay for up to a month in the mouse brain. So a lot of people find that quite exciting because it's very long-lasting structural modification in the brain. Yet the time scale, seems to match or in alignment with some of the clinical kind of the long-lasting efficacy. So in some of these recent study, we really want to follow up on that finding and ask a couple of questions. One is, as we just discussed, there are many different cell types in the brain. So which particular cell type might be undergoing plasticity? And then in the other study that you mentioned in cell, we were very interested in, okay, you have new connection, but what specific circuits are those? Because we have a synapse, but then you must have an input and an output, but where do they come from? Understanding not just more or fewer synapse, right, but exactly which pathway is being modified is critically important because it can inform us, again, which input are being strengthened or weakened, and then give us a lot more quantitatively how the circuit is being modified. So I would consider, yeah, these to be, asking just more specific question. Okay, we have more plasticity now, but where are they occurring? So that's sort of the impetus for these new studies.
So, right. So when I think about formation of a dendritic spine, which is a potential synapse,
Yeah.
um, I think about the cytoskeleton, the polymers of actin in particular that uh, allow the neuron to grow.
Hmm.
And so spine formation, my understanding is involves polymerization of actin, and so they kind of get pushing out, I guess, of the membrane and formation of a spine. And then there has to be some kind of stable, long-lasting changes that allow it to maintain its structure. Um, so you've done right, and calcium is probably a very important trigger. So, for example, I mentioned I was studying showing that glutamate uh, promotes synapse formation. The way that does it actually is it acts on glutamate receptors, causes calcium influx locally,
Hmm.
and then you get uh, protrusion of it's almost like a filipode on a growth cone. That's very narrow
Yeah.
projection. And then there's further interactions between the pre and postsynaptic cell that lead to the formation of synapse. And if you block the calcium influx, you don't get that. So, um, yeah, I would assume that there's some role for calcium in this process. And so somehow, if we assume let's just assume this, it may not be the case as you pointed out, these structural changes, but let's assume that the psilocybin is acting on the 5-HT2A receptor.
Yeah.
And that's coupled to a signaling pathway that, well, it does a couple things. It activates a kinase uh, uh, and protein kinase C, but it also causes calcium release from these internal stores in endoplasmic reticulum.
Yeah.
So would you predict if you block protein kinase C, if you block uh, whatever IP3 receptors, so block calcium release from
Yeah.
the endoplasmic reticulum in those neurons that you're looking
Yeah.
at the spines,
Uh-huh.
that you would no longer see these structural changes.
Yeah, so we're fascinated by this process, right, of the spine formation and looking at differences between like drug evoked versus kind of um the canonical or what we know happen in a typical brain without the drug. Some things we know, and I think your intuition is correct. For example, we have done calcium imaging in the dendrite before and after drug and compare. And when you do that, you can see elevated number of calcium transient up at the dendrite and in the spines after a single dose of psilocybin or ketamine. Um, so definitely the drug increases the amount of calcium in those dendritic compartments. We have also done knockout experiments where we can completely remove the serotonin 2A receptor in the dendrites, or in the whole cell actually.
Uh,
In the pyramidal cell, they are known to be dendritic localized. And when you knock out the receptor, then in the postsynaptic cell, then you no longer have these structural remodeling. So there are some key elements where you know must play part. In terms of where that calcium come from, I think there could be two places. One is, like you said, maybe through the G protein couple receptor pathway and then internal store release. we haven't tested that one as much. There's another possibility, which is, these again are GQ couple, which means they excite the dendrites on its own as well and changes the memory potential. So it's also possible that by changing the memory potential, that also just have more external calcium flooding in. So that part we haven't quite teased out, but I think it would be quite interesting to see. And they have different ramification in terms of what it means for how the drug work. Yeah,
Now, in the depression field, in terms of mechanisms, there's been a lot of data and emphasis on brain-derived neurotrophic factor, BDNF.
yeah,
Um, and I think rightfully so. It seems like all of the antidepressant treatments that are effective, even if you go back to electroconvulsive shock therapy, the psychedelics, physical exercise, which is actually an antidepressant,
mm,
they all, and even the the most commonly prescribed SSRI increase BDNF levels.
yeah.
Um, and do you think, well, this again, intuition, there's probably a role for BDNF in what you're seeing.
Yeah, I think for sure at the permissive level, I think there's a good amount of evidence in terms of even going back to the 90s, one of my old Yale colleague, Ron Duman, passed away sadly, but he has some early work showing that if you give a single dose of psychedelic, in that case he used DOI, you can see increased levels of BDNF in the I think rat cortex when he was doing it. So there's some really early evidence showing centrally there's BDNF associated with these drugs. And of course, he has a lot of other work on ketamine beyond psychedelic. Um, uh, so I think, yeah, like the central role in terms of the neurotrophic factors probably is part of the story. What's interesting is I think recently the field has debated now whether it's sort of a permissive role, maybe downstream of the serotonin receptor activation, or maybe there's some suggestion that maybe some of these drugs could even directly bind and interact with the TrkB receptor. So that remains debatable, I think. I don't know if you follow that, but there's some people suggest it could directly interact with the TrkB receptor. There are other people suggest maybe it does not. I think there's recently a paper showing that maybe it does not really bind. Um, so I think the details remain to be seen, but for sure, I think neurotrophic factor is part of the mechanism. Yeah.
Now, when you give psilocybin to a mouse,
Hmm.
unanesthetized mouse, uh, are there acute behavioral changes that it causes that can be quantified?
The most obvious acute changes is the so-called head twitch response. So, which means that they move their head in a sideward motion, and they do it exceedingly fast. They only do it a few times, but if you look at the frequency, it's 90 times a second. So it's a really quick head movement. So in the lab, we usually monitor it with a high-speed camera, this head twitch response. It comes on very quickly after you inject the mouse with a single dose of, for example, psilocybin or other kinds of classic psychedelic, and then also wears off quickly. So it's a acute behavioral effect. It's a very interesting effect because it's very species specific. It's in mice and then definitely in rabbits, but only in a few species. In human, we take psychedelic, we don't twitch our head. So it's a very mouse specific behavioral effect. But it's very reliable. So it's really been used as a way to study these drugs for drug development purposes.
So there's no behavioral response that one might hypothesize would be similar to hallucinations in humans.
I think it's tough to know. That's, I think, very active area of research because in human, you can talk to them and say, hey, how do you feel and what do you see? And then they will describe to you these very vivid imagery. But in a mouse, you can't ask a mouse what they saw, right? So, and it's somewhat hard to assay because the perceptual changes could also be quite vary. Even in humans, it's quite vary. In a mouse, at least the classic work, there hasn't been any kind of task that I think confidently pull it out. I know there are some research lab working on this to see if they can have a visual task to ask the mouse if they're seeing something. But right now, it's still kind of hard to prove.
Yeah.
whether the mouse is truly tripping like us.
And is it known which circuits are mediating the head twitch thing?
No, I think that's another mystery. Even though the head twitch is known to us since the 1960s or so, nobody have really isolated the location of the head twitch response. One thing that's interesting is it doesn't quite correlate with the amount of drug in the brain perfectly. So if you give the mouse a dose of psychedelic, the head twitch response come on very quickly. It max out at about 10 minutes. But the drug itself takes about 30 minutes to peak in the brain. So there's a mismatch between the timing, which is also interesting. To me, again, just because of how fast it is and how in it the motor response is, it suggests something a bit more peripheral, maybe like hypothalamus or
Yeah,
brainstem.
yeah.
But that's also just my opinion. And other people think maybe it's cortical, but nobody has really nailed it down.
Okay, then what about non-human primates? Uh,
Non-human primate, yeah, I think they're a bit closer to human. So that's also interesting. In the older reports, when people give, like, say, LSD to a macaque monkey, they would report things like the monkey spacing out, the monkey grabbing things in the air.
ah,
But those again are in the 60s and 70s, where back then the researcher there's no video record of it that I could see, and then they would just write out the description, and it's usually like one or two monkeys. So it's depends a bit on interpretation sometime. Yeah, so I would love to see some of those type of experiment repeat now, with like quantifiable video analysis. And I think people are doing that and finding similar things where they do exhibit behavior which we would much closely associate with actually some kind of perceptual disturbance in the non-human primates.
and are you going to or started to integrate optogenetics into your repertoire of approaches to, uh, I don't know, help you further dissect what's going on?
We have in one experiment, so optogenetics, allow us to stimulate neurons and drive them to fire or inhibit neurons. In a couple of recent study, what we've used optogenetics is a way to identify the cell types. Again, we're very interested in how different cell type, like pyramidal cell, interneurons, how they respond to psychedelic. And then when we do electrophysiology, it's almost blind and we don't know what cell type we're recording from.
Yeah.
So one way we view optogenetics is we have these specific cell type express channel rhodopsin, and then after the whole recording, we would shine light on them, and then those specific interneuron would fire, and then we can then tag them and know that oh, aha, we recorded from this GABAergic cell, or aha, we recorded from
Huh.
this
Yeah.
PD pyramidal cell. So that's been quite helpful in terms of dissecting the different cell type and how they respond to psychedelic drug action. So that's
Hmm.
one place where we've used actually optogenetics quite heavily, yeah, combined with electrophysiology.
How big is your lab now? How many people working in the lab?
We have at the moment 8 people in the lab. We have 3 postdoc and then 8 graduate students.
Wow.
Sorry, 5 graduate students. A total of 8 people. 3 postdoc, 5 graduate students.
That's a
Uh,
lot of graduate students.
yeah, the lab changes over time. I mean, I think when we were at Yale, we were a little bit more postdoc heavy. Now that my lab has moved to Cornell, here I think Cornell has a fantastic graduate program. We have a student from the neurobiology program, a couple students from the biomedical engineering program, and then a couple students from the molecular biology graduate program. What's exciting about working on psychedelic drug action, it seems like even though these people come from different disciplines, they all are very aware of the excitement with this line of research, both in terms of the translational impact, but also just the opportunity to find something new, fundamentally new about the drug action. So we have a lot of interest in terms of people who want to study this using different approaches.
Yeah, it's always good to have the postdocs can kind of mentor graduate students on a day by day basis, right? And are you spending, do you have time to spend in the lab actually hands-on still?
So my contribution have actually been more on the analysis side. So yeah, it is getting harder now with a lab of that size to
Yeah.
go in the lab and do the hands-on stuff. I do sometime like when because I have optics background. Sometimes when the microscope is misaligned or maybe
Uh,
the laser doesn't turn on, they would ask me to go do it because I have, I was trained to do that. But most of the time, when I do lab work now, I actually do some data analysis. Um, One thing actually I'm very good at is programming. When I was young, if I were not to become a neuroscientist, I probably would become a game programmer. That was something I was very keen on as a teenager, like playing games and then programming games. And uh, so I'm very good at programming and some of the data, labs data, I've analyzed them, write code to do this kind
interesting.
of analysis. And it's
Interesting.
becoming more and more needed, right? Because the kind of imaging data that we get, electrophysiology data, is just such large scale. These days, a lot of times you spend one day recording from the animal, you get hundreds of neurons, and then you spend like the next week basically analyzing the data.
Yeah.
So I try to help in those regards. Yeah.
My understanding is that the coding, yeah, there's some universities actually in Baltimore that kind of, a lot of them have like really built up their computer science program and so on, and people learning coding. But my understanding is artificial intelligence technology is going to be able to do a lot of coding, or maybe already is. But
Mhm.
wh what do you think about, have you thought about, I'm sure you have, uh, AI applications to your own research?
Yeah, that's something I think about too. I think AI can be done in different ways. One way it has not influenced me yet, is I have not used it to actually code. I know some people have it kind of code side by side with AI. At least for me, because I have some programming background, I find myself code pretty fast. So I haven't tried to
Ah,
incorporate AI as a way to code because I also don't like reading other people's code. So I don't know how I would like have to go through AI's code. Sometimes it's hard to read other people's code. But I should probably try it anyways. I'm just a bit resistant and older.
well, and
One thing that AI has helped, that I would say, is that we have incorporated some AI algorithm to do a lot of uh, object detection, like cell counting. So, for example, we have these large scale, as I mentioned, brain-wide imaging of immediate early gene with labeled nuclei. And then we recently have a paper, we do these rabies viral tracing where they express the fluorescent protein in a cell. And this label 100,000 cell in the mouse brain, the whole mouse brain. I mean, nobody's going to go and count them individually. So we have AI that do object recognition and basically count them and locate them across the whole mouse brain.
one
Um,
of
so,
one of the one of the investigators in my laboratory when I was at the National Institute on Aging was Henriette von Prague, and she did. She was the one when she was with Rusty Gage at the Salk. She showed that exercise stimulates neurogenesis in the hippocampus.
mhm,
And then when she came to NIA in my group, she, as a tenure track investigator, she used the rabies virus to determine which neurons are providing inputs to the newly generated neurons in the hippocampus. It was
mhm.
pretty interesting, the kind of the timing and location of the inputs over time after these new neurons are being
Mm.
integrated into the circuit. So that's you're using rabies virus kind of same kind of way to determine connections.
Yeah, that's correct. So, as I mentioned, one of the more recent study, we were very interested in input, and then that's exactly what we use the rabies virus for, where we know that if they grow a dendritic spine, there must be an axonal input. But in the frontal cortex, you have like more than 20 area to send a lot of inputs there. I mean, how do you test every single one of them if you image them one by one? So we use the rabies virus, which I describe to people as almost a bit like the Google self-driving car and then mapping the roads. I think it's a bit like that, right? You put them in the brain, these self-driving, well, rabies virus, and then they trace all the connections for you, kind of like how Google generate a map. So in this way, we, did this mapping with rabies virus for mice that have injected with the psilocybin virus, psilocybin drug, and then also in mice that receive vehicle, and then look at basically how those map differs, the connectivity differs between the mice that receive the psychedelic or not. And
I'm trying to remember if I can't. I may be getting mixed up. My younger brother is a veterinarian,
Mhm.
and he's he's got rabies. Is it a vaccine or shot? To prevent rabies. The virus. I can't remember whether the virus Henriette used if they had to get rabies shots. It's not. Is it pathogenic? I can't remember.
Well, normally it's extremely pathogenic, but the one that we use was the one developed at Salk
Yeah, yeah,
by people like Ed Callaway. So
yeah,
they engineer it to remove one of the component called G protein, so it's can no longer replicate.
yeah,
But then it was quite ingenious because you can then put that back only in some neurons, so it just jumped one synapse and then stopped right there. So we use a version of this rabies virus. It doesn't spread the whole brain; it just
yeah,
spread one synapse.
yeah.
And then we were able to map again the connectivity change with or without drug. And one thing that kind of surprises was with the psilocybin was in terms of the changes in the connectivity, it was somewhat global. Like we were initially going in thinking maybe the drug would alter one or two pathway, but it turns out that there are quite a lot of pathway that get changed in the brain. But there is a pattern to it, and it's very network specific. So it's not a random change, but it does involve much of the brain. And then depending on which network and which region it come from, it can either be strengthened or weakened. And this seems to happen in a mouse sort of within a week after the drug was injected.
One general idea I think I remember is that the psychedelics kind of disrupt communication between your normal sensory inputs and your ongoing thought processes. So there's a so what are your thoughts on that? You start out as interested in visual pathways, I think you mentioned,
Yeah.
and in humans, a lot of the psychedelic experience is visual. Um, so these are clearly not visions coming through your eyes; they're internally generated visions. So how do you look at that, and how might your work help tackle that question?
Yeah, so there are different theories for why the visual hallucination arises. One theory, mostly from human study, is that maybe the visual cortex becomes more active. That's why things become more vivid. But I'm actually more partial to a different theory, which is that maybe the visual cortex actually receive less input and is less active. So the rest of the brain actually then fill in with its
Yeah,
own information.
yeah,
And there's some evidence suggesting this. For example, if you go to a deprivation tank and you shut yourself out and you're in complete darkness for, several hours, you can start hallucinating just from the lack of visual
wow.
input. And then when
And then
people
at night you dream.
And then, yeah, and then you close your eye and then you can
Yeah,
dream. So there are a lot of suggesting that the lack of input can induce hallucination in other kinds of contexts. And then some older monkey work also show you put an electrode into LGN or V1 of the monkey, you can also see reduced activity. Uh,
hmm.
so yeah, so I'm more partial to that idea. We haven't explicitly tested, but I think it would be a great research question to, look at it. For example, use the mouse as a model where you can target these pathways quite specifically, look at the transmission of information up from the thalamus into the visual cortex, and then maybe look at some feedback back in from the frontal cortex and see if it really support this idea that is a reduction in the sensory information and maybe a further reliance on some of the prior belief that drive the hallucination.
Yeah. What's going on? And there's a lot of clinical trials going on with not only for depression, anxiety disorders, but also addiction, I think. Um, so you mentioned one of the regions you focused on, apparently because the neurons are lighting up a lot in terms of their activity when you give
Mhm.
psilocybin, the cingulate. Um, so my understanding, the cingulate cortex, well, if you look at like the brain, cut it down this way and look there, it's the kind of wraps around the corpus callosum,
Yes.
right? And so it's essentially physically under a broad scope of the overlying cerebral cortex. And if you just look at that kind of like, oh, well, then maybe this brain region's really important in somehow coordinating activity between different cortical regions or between communicating from cortex to lower brain regions. Um, so let's just hypothesize that everything you're seeing is due to effects of psilocybin on neurons in the cingulate cortex. Is there some way you could imagine that could be in terms of the way this known circuitry is organized?
Yeah, I think there's some aspect of the circuitry is interesting that may be consistent with the drug action. I don't want to say explain it because I think that requires a lot of evidence.
Yeah,
But as you mentioned, the cingulate, we study anterior cingulate, which would be like the front of that cingulate. There's also posterior cingulate, which in the mouse we've also looked at called retrosplenial cortex. And then a lot of people think that's sort of the seat of the so-called default mode network, which would be kind of
yeah,
how people think of themselves in terms of their ego. And our studies show actually with the rabies tracing that the connection that go from this area that's more like the homolog or default mode network going up to the frontal area and then coming back down, that loops tend to weaken. So I mean, one can speculate what that means. I mean, but then it is interesting example and demonstration of a weakening loop that involved the posterior cingulate portion. That was one of the notable effect of the drug. And then the other thing that we found was that out of the cingulate into the subcortical area, so this is through the pyramidal tract of the brain, that part actually seems like it got strengthened sensory input. So they're also consistent idea that a long-term effect of the drug is to, take in some of the sensory input and then right away routing them to subcortical action.
huh,
So I think, yeah, we're beginning to see some circuit changes that might relate to the drug action, and maybe certain pathway like these could be mapped onto different aspect of what the drug does. I think it's still you need to then go in and test, manipulate these areas and see these pathways and see if it map on a drug action. But seeing these changes is quite interesting, and they're quite specific.
in humans, what's going on? So the functional brain imaging, functional MRI in
Hmm.
humans, it's valuable, but it's not very. The spatial resolution is poor. You can't look at individual neurons. You can't even really, I don't know what, very the size of the pixels are. There's probably hundred, tens of thousands of neurons, and then also the time resolution isn't very good. But is there anything going on in well, looking at the human brain and trying to get at some of these questions we asked, or that we've talked about, and that you're trying to get to in the animal model, where you can apply all these techniques that you can't do in humans? What's going on besides clinical trials in humans?
I think that would be very valuable. At the end of the day, a mouse brain, is not does not equate to a small human brain. I mean, they're different brains.
Yeah,
For example, I don't think the mouse really have a dorsolateral prefrontal cortex. There's some brain region that might even be missing and sort of specialize in human and or other non-human primates
yeah,
that the mouse lack. Um, so I think it's really important to try the two. I think there are two things in human that excite me in terms of biological research for psychedelic. One is, yeah, using neuroimaging to track connectivity, but maybe do it longitudinally. So I know that there's some people now really doing dense sampling, imaging multiple times before, and then maybe even during, and then afterwards, and then look at what they call like precision neuroimaging to see how the connectivity change in the same individual over time. So that to me have quite a bit of value and be very interesting to see if some of our rabies tracing result map onto some of those functional connectivity readout from humans. And then the second place where I see, and I use that information a lot from human is it's also possible to use PET imaging to look at how the drug engaged the serotonin receptors in the brain.
Huh.
So how much of the receptor is bound by the drug. That can be done in a live human. So there's a lot of value, I think, to relate the potency of the drug in terms of uh, what level of drug engagement do you need to start having these perceptual changes or to start having maybe even therapeutic effect. Because ultimately, yeah, I mean, these are drugs, so we need to know how
Okay.
much they engage the correct target. And that can be done in human. And I think to translate correctly, we need to know that receptor interaction. That's another thing that I think human can study, biological study can provide a lot of value.
All right. So that's already being used. So PET imaging. So they have uh, essentially radioactive psilocybin, radio labeled psilocybin, and then give it IV or something, and then look where the relative amount of radioactivity
Yeah.
in the brain. Is that what's going on?
Usually I have something else that's radioactive that bind to the same target, like the
Same
serotonin
receptor. Yeah.
receptor. Yeah, so it's
Yeah.
a competition doubling, but it's similar idea. They would either just give the tracer to normal people, in which case it would bind a lot, or you can first give them psilocybin and then give them the tracer
I see.
because psilocybin is already there, then the tracer cannot bind and
I
then a
see.
reward signal. So then you can see how many of the receptor was already occupied.
Huh. Yeah.
So that study has been quite useful. So, example, from those kind of study, we know that you need a drug that occupy probably around 60 to 70% of the serotonin 2A receptor in a human brain to have a perceptual experience. So you do need a certain amount of drug and
Yeah.
engagement. So if you have too weak a drug or maybe too weak like a partial agonist, it might not do the trick. So it's important to know yeah what where you start to get the behavioral effects.
Yeah. Has a therapeutic. Yeah, they must have done some like dose response stuff early on, the Hopkins group or other groups, uh, other groups doing that. Um, so I guess, from the common experience, about uh, a bad trip versus I guess, a pleasant psychedelic trip. The idea if you take a high dose of LSD or psilocybin, then your experience may be not as good as if you take a lower dose. And so I guess if you completely uh, if you activate all the 5-HT2A receptors, is that worse or equivalent or better than occupying, say, half of the receptors with psilocybin?
Yeah, dose definitely relates to the intensity of the subjective experience. I think at the lower dose range, people describe more like some kind of distortion in the perception where maybe like the grass look greener or like a straight edge look a bit rounder. And then at higher dose, as you're alluding to, maybe you can start to actually see hallucination properly, like actually objects that does not exist. Um, in terms of having a positive or negative experience and how anxious, that also I think relate sometimes a lot to the environment and the setting.
Yeah.
So I think it's actually seen a lot less, as far as I know, or reported in the clinical setting where it's quite controlled, where you have a room, it's a very safe environment because you have
Yeah.
usually two medical professionals there. They have certain lighting, certain music, that everything is sort of again regulated.
Yeah.
My understanding is it's mostly either neutral or positive experience, and there are very few what people so-called bad
Yeah.
trip.
Yeah.
If you have difficulty, the medical professional is there to help you. Versus yeah, setting where it's more recreational, which would be quite variable, yeah, in terms of um,
Yeah.
what the experience could be. So I think that part is also interesting and it's quite uniquely human. I don't know if one can study that kind of process in in animals, that very context dependent aspect of the drug action. But it is a very fascinating part of of psychedelics.
Yeah.
Yeah.
Okay, Alex. Well, uh, thanks for taking the time for this. I enjoyed talking to you and I look forward to seeing, how things proceed moving forward. That's yeah, the general public's very interested in this. Washington, D.C. decriminalized mushrooms anyway, psilocybin mushrooms, Oregon, uh, and I think in Maryland here,
Colorado. Yeah.
Maryland, where?
Colorado, also
Oh,
one of
Colorado,
the
yeah,
early places with clinics. Yeah. Yeah.
yeah. And so we'll see what happens. It's certainly, yeah, my own impression is that's not going to cause a lot of problems. It's certainly not, these drugs aren't addictive. And, that's one really important aspect of them. They have the side effects seem to be, if any, not, hardly any or non-existent. Uh, one thing is that there's companies now, and I don't understand this too much. I think it's just a money-making thing. There's a number of companies I'm aware of that they're essentially taking psilocybin and modifying it, doing medicinal chemistry, change the structure of it a little. And then the idea is in the future they'll be able to get that into the clinic or something. But it seems I don't see how you can improve on
Oh,
what's already
well,
there. Yeah,
yeah, I mean I take a different view there. I mean, I think well there are a couple of things there. One is I think psilocybin or other 5-MeO DMT, there's a few of these psychedelic that come from nature. If you look back at the history of drug development, we have other things like that
yeah,
too.
yeah.
Yeah, but over time, we do, again, through chemistry, can modify it slightly to reduce certain side effect, or maybe, have the drug act a bit longer. Over time, that has again happened a number of times, leading to again things like aspirin, for example, right, come from like a willow tree. But then over time, you can make it better. So I think I don't see why psychedelic would be an exception to that. I think there are probably ways to engineer to make it a little bit better over time. And then the other effort that maybe you haven't mentioned is like there's also a big effort to maybe do big modification to try to take out the trip, but then keep the therapeutic part intact. So that I think remains to be seen whether you can have a drug that act on the 2A receptor, does not cause the hallucination, but maintain the
Uh,
therapeutic effect. But if that works, we don't know if it will. It can open up, I think, access, which would be useful. Right now, one of the impediments for doing these kinds of treatment is you need to be in a regulated environment. So you need to go into the clinic beforehand and then during, while you're under the influence of the drug, and then usually afterwards in integration. That all take many, many hours and it's quite expensive. So if some of the acute effect can be removed or even reduced, I think that could make it more accessible and hopefully lower the cost of these kind of treatment.
yeah,
So we'll see. We'll see, I think, how it goes. But I think I'm hopeful that over time we'll have improved versions of these compounds,
yeah,
at least in terms of therapeutic improvement in terms of the therapeutic part.
yeah.
Yeah.
Okay, Alex. I hope winter gets over pretty soon, but it looks like it's going to get a little colder and snow's on the way. So, um, Ithaca's a nice place. My daughter and her boyfriend actually go up in that area It's kind of one of their favorite places to go on vacation, drive up there from down here in Maryland.
Mm. Yeah, it's beautiful up here. Winter is good too. I mean, it has its own charm.
Yeah. Okay, thanks, Alex.
Thank you. It was great talking to you.
Yeah, bye.